Strepsiptera stylopization. This order of insects has a few hundred species that display a prominent sexual dimorphism in adults. The males have branched antennae, large eyes, paddle-shaped mesothoracic balancers, and large metathoracic wings. Females are apterous and larviform. In all families except the primitive Mengenilidae, adult females that have been described lack legs, are not able to move in an orderly way, and never leave the body of the host. First instar larvae, called triungulinids, are of the planidium type and bear a close resemblance to those of Rhipiphoridae and some Meloidae. The order is considered by some taxonomists to be closely allied to Rhipiphoridae, and the free-living females of Mengenilla and Eoxinos have a resemblance to the aberrant genus Rhipidius. Larval development is completed within the body of the host in a way similar to that of Rhipidius in cockroaches, while the Rhipiphoridae attacking Hymenopterous larvae are external except for a time during the first instar, Westwood 1836, 1939, Clausen 1940. Because of their small size, great activity and short life, adult males are difficult to collect. Many species are thus described on the basis of one sex only. The definite association of adults of the two sexes of a species is possible only by rearing, preferably from the same host individual or from those of the same colony. The members of the order have come to be known under the common name of Stylops from the original generic name of the entire group. Clausen 1940 noted that a parasitized host is said to be stylopized and parasitization by a Strepsipteran is referred to as stylopization. Hosts of Strepsiptera. Strepsiptera parasitize four orders of insects: Orthoptera, Hemiptera, Homoptera and Hymenoptera, with the latter including the largest number of families. Records of attack on Orthoptera and Hemiptera are few, while both the Cicadelidae and Fulgoridae frequently are hosts. However, the Hymenoptera, particularly families Vespidae, Eumenidae and Andrenidae, are most frequent hosts. Extended early host lists were presented by Pierce 1909, 1918, Robertson 1910, Salt 1927b, Salt and Bequart 1929, and Ulrich 1933. Generally, the hosts of the different families, based on the classification and records given by Pierce 1909, 1911, 1918, are as follows. Family: Mengenilidae, Mengidae, Stachotrimatidae, Calopharyxinidae, Myrmecolacidae, Stylopidae, Hylecthridae, Xenidae, Triosoceridae, Halictophagidae, Alenchidae. Hosts: unknown, unknown. Orthoptera, Locustidae, Hemiptera, Pentatomidae, Hymenoptera, Formicidae, Hymenoptera, various families. Hymenoptera, Hyliidae, Hymenoptera, various families. Homoptera, Cicadelidae, Homoptera, Cicadelidae, Fulgoridae, Homoptera, Cicadelidae, Fulgoridae. Pierce 1909, 1918 believed that each Strepsipteran was confined to a single host species, and each tribe to a family, although recent studies show this to not always be the case. Clausen 1940 believed that host specificity was not usual in this order. For example, Bahart 1936 found that many species of Stylops are parasitic in two or more similar species of Andrena. Generally the order is regarded as beneficial primarily because of attack on Cicadelidae and Fulgoridae. Muir 1906 found more than 70% of Perkinsiella vitiensis Kirk. On sugarcane in Fiji were parasitized by Elenchoides Perkinsi Pierce, equals Elenchus tenuicornis Muir, and 30% of Idioceras atkinsoni Leth. In India contained Pyriloxenose compactus Pierce, Sebramanium 1922. Schrader 1924, in studies of Xenos, Acrochismus, Wheelery Pierce as a parasitoid of Polistes, found that the number at first attacked in a nest is low, but that this finally builds up until it causes almost an extinction of the colony. Peel 1933b reported parasitization of 25% of Spex nigellus Smith by Ophthalmoclis sp. And Theobald recorded 50-70% parasitization of Andrena by Stylops sp. Honeybees are rarely attacked, Beljovsky 1926. The highest and most consistent parasitization was recorded by Kirkpatrick 1937a, b. per Corioxenose antestiae Blair on the pentatomid Antestilinidicalis stall, a pest of coffee in East Africa. The portion attacked ranged from 12-84% during the year, with an average of Gt. 40%. In Hawaii several species from various parts of the world were imported for the biological control of sugarcane leafhopper, Perkinsiella saccharicida Kirk, but none were established. Clausen 1940-1962. Biology and behavior. Early detailed accounts of the biology and behavior of Strepsiptera were by Nasanov 1892, 1893 Bruce 1903, 1905 Perkins 1905 Hoffender, 1910 Pierce 1909, 1911, 1918 Wheeler 1910, Smith and Ham 1914, Schrader 1924, Schultz 1925, Ulrich 1927, 1930, 1933 Parker and Smith 1933b, 1934, and Kirkpatrick 1937b. Cooper 1938 gave an extended account of the internal anatomy of the immature stages and adults of C. antestiae Blair. Reproduction. Strepsiptera are larviparous. In view of the manner of development and the position of the adult females, it is improbable that any species lays eggs. There is a uniformly high reproductive capacity, which is essential because of the hazards encountered by the triungulinids before they reach the host. Newport 1845-1853 recorded rearing more than 7,000 triungulinids from a single female of Stylopsiptera manup. Pierce, 1909, 1911, 1918 counted 2,252 in one female of S. Swanky Pierce. Xenos wheeleri, X. oriferi Pierce, and Eoxenos labalben and Pyriloxenos compactus have over 1,000. Clausen, 1940-1962. Studies on oogenesis and embryology were made by Bruce, 1903, 1905 Hoffman, 1913 Schrader, 1924, and Noskovich and Polishinsky, 1924, 1928. The latter gave a brief account of supposed polyembryonic development in a species of Halictoxenos parasitic in Halictus simplex per. The brood of a single female emerges from the body in a short period of time, usually all in a single day, and several thousand triangulinids of E. labalben emerge from one female in less than 1 minute. Parker and Smith, 1933b, 1934. Sebramanium, 1922, 1932, reported that the triangulinids of P. compactus and Tridactylophagus mysorensis suber shot out into space, through the opening on the ventral side of the cephalothorax. Exiting from the body of the parent Eoxenos is through the genital opening on the venter of the seventh abdominal segment, while in forms having opodous females the triangulinids at first pass through the genital ducts into the brood chamber and from there to the outside through the genital opening between the head and thorax. Many of the triangulinids of Coreoxenos emerge through the oral aperture of the head. Kirkpatrick, 1937b. The membrane covering the genital opening is perforated at the time of mating, and this provides the normal point of exit in most species. Clausen, 1940-1962. Clausen, 1940, pointed out that the use of the term larva position in respect to reproduction in Strepsiptera is inexact, for it implies an act on the part of the female. The triangulinid leaves the body of the parent female entirely of its own accord, unaided by any stimulus of the female. The simultaneous development of all eggs within the female's body requires the emergence of the entire brood of triangulinids. Although this is true for the majority of species, a markedly different behavior occurs in Coreoxenos antestiae. Here females have been found to produce young over a period of more than 3 months. One individual produced 3,720 in 95 days, with 134 remaining within the body at death, and another 2,220 in 49 days. In the laboratory, a female produced an average of ca. 50 triangulinids per day. Generally, females of this species continue to produce progeny until the host dies. Despite the protracted reproductive period, only a single mating is necessary during one lifetime. No instance was found of spermatozoa being present in the body more than 18 days after mating, and thus embryonic development may cover a minimum period from fertilization to emergence of 6 weeks and a known maximum of ca. 19 weeks. Kirkpatrick, 1937b, Clausen, 1940. Triangulinid larvae. These larvae are thought to leave the maternal host in a relatively short time, particularly if the host is a homopteran. The jumping habit of the triangulinids, which is common to the Planidia of several orders, was first observed by Saunders, 91853. In E. labalben jumping is in an unusual way. The abdomen is raised from the substrate and the caudal cerci are brought forward beneath the body, after which the body is suddenly straightened to project the insect into space. Triangulinids of several species are believed to be positively phototactic, this in order to aid in attaining a position in which contact with a carrier or host may be made. It has been shown in Coreoxenos that little movement of the triangulinid occurs, and apparently no searching. Thus, the triangulinid is dependent on direct contact with the host. The alert or waiting position, which may be held for hours, is characteristic, the terminal cerci being bent forward under the abdomen and the anterior end of the body considerably raised and supported only by the hind legs. This is in contrast to the habit of the Planidia of other orders, which stand erect on the caudal sucker, with the body braced only by the caudal cerci. Clausen, 1940. In Coreoxenos, jumping occurs as a result of the stimulus provided by a nearby moving object, and especially in response to certain colors. Colors most attractive are black, red and orange. Kirkpatrick, 1937a, black and orange being the predominant colors of the normal host, Antestia. If the object to which the triangulinids become attached is not an immature Antestia, they leave at the first opportunity, while if it is an Antestia they make their way to the dorsum of the thorax and abdomen, between the coccyx or between the head and thorax, where they cling immobile, with the mouth parts and legs, until the host molts. Penetration of the host by this species usually occurs at the time of the molt, yet it has been shown that triangulinids will quickly enter adult hosts bearing mature Coreoxenos of either sex. Entry is effected in such cases at the point of extrusion of the older parasitoid, and it is probable that a second generation of parasitoids can develop to maturity in Antestia if that number of individuals is not so high as to cause premature death. Clausen, 1940-1962. Strepsiptera developing in Homoptera reach the host directly, without a carrier, for the triangulinids are released on the foliage visited by nymphs and adults of the host. These insects are usually so abundant that the chance of a triangulinid reaching a host directly is great. With hymenopterous hosts the aid of a carrier is advantageous, though possibly not essential. The larva within the cell, rather than an active stage of the host, must be reached. Bees and wasps that are subject to attack have two general feeding and brood-carrying methods, which have a bearing on the chances of the triangulinids to reach the larva. First, the food is stored in the cell, the egg laid on it and the cell is then sealed, or secondly, the egg is first laid in the cell and the larva is fed periodically until maturity, after which the cell is closed. In the first case, the period in which entry into the cell is possible is very short, probably not more than 1 In the first case, the period in which entry into the cell is possible is very short, probably not more than 1 day, and thus such species are most likely only infrequently attacked. However, this omits the possibility that the triungulinids may be able to penetrate the cell after it is closed. If entry is on the day the egg is laid, the triungulinids have to wait for some time until the egg hatches and the larva is sufficiently developed to withstand attack. Because of the small size of the triungulinid with respect to that of the host, its penetration into the first instar larva seems to be without serious injury to it. In the second case, the cell remains open throughout the larval feeding period, and the larva is tended 1 or more times daily by adults, so that the opportunities for parasitization are greater. Clausen 1940-1962. Although undoubtedly some triungulinids transfer directly from the maternal host in the nest, this is not thought to be common, for parasitized females do not engage in nest building, at least not at the time that the parasitoid brood escapes. Pierce, 1909, 1911, 1918 considered that transfer was effected mainly on blossoms and at other places frequented by bees and wasps. Westwood, 1839, first suggested that transfer took place on blossoms. With hosts of social habit, there is an excellent opportunity for transfer by direct contact in or near the nests, in contrast to the improbability of such occurring among solitary species. Clausen 1940-1962. Penetration of the host is always in the first larval stage, and advanced triungulinids of several species have been dissected from Hymenopterus larvae. They seem to lie free in the body cavity. Saunders, 1853, placed triungulinids with almost fully grown Polistes larvae and observed that penetration occurred soon thereafter. Dissections a week later showed the larva just undergoing the first molt. Kirkpatrick often observed the entry of Coreoxenos into its pentatomid host, this usually taking place on the pronotum at the time of molting and is effected within 20 to 30 minutes of the beginning of the molt. Although entry may occur at any host molt, it is most successful at the first 3. Surprisingly, the newly hatched nymph is never attacked even though in physical condition it is much the same as at the time of following molts. Certain deformed antestia are apparently immunity to parasitization by Coreoxenos. A portion of the older nymphs are found with malformed antennae, and rearing experiments where these were subjected to attack by triungulinids of the parasitoid showed that few were able to enter the body. Kirkpatrick 1937a, b. Only 18 of 54 triungulinids entered the bodies of these individuals, and only 8 of 49 hosts were parasitized. This is in contrast to results secured with normal hosts, among which over 90% were successfully parasitized when exposed in the same manner. A deficiency in the molting fluid, resulting in a difficult molt, may be responsible for the malformed antennae. If this fluid provides the stimulus for penetration by the parasitoid, as is probable, then such a deficiency would reduce the stimulus to the point where triungulinids would not react to it. Clausen 1940-1962. Development of larva. In C. antestiae, if penetration of the host body is in the thoracic region, migration of the parasitoid larva to the abdomen usually occurs late in the second stage. From then on, there is little movement until that incident to the extrusion of the cephalothorax. In Hymenopterus hosts, the larvae also migrate to the abdomen during the early period of development if entry has occurred in the anterior portion of the body, and probably while the host is still in the larval stage. Early researchers found only 3 larval instars, but later studies found that there are probably 7. Schrader found that males have only 6 larval instars. In molting, a transverse break occurs across the anterior end of the body, and the larva wiggles out of the exuvie. Feeding is mainly by diffusion through the integument, although in Coreoxenos movements of the rudimentary mouth parts were observed, but it is not certain whether this represented direct feeding. Smith and Ham 1914 examined the alimentary system of S. melitae kirby and found it to be degenerate and functionless. The larva may be determined to sex at a comparatively early developmental stage, especially in the 3rd instar. Extrusion of the cephalothorax from the host body occurs in both sexes during the 7th stage. Species attacking Hymenoptera extrude the cephalothorax during the pupal stage of the host or after it has transformed to the adult stage but before the integument has hardened. In pentatomidae hosts this occurs only after the adult stage is attained, but in the Homoptera it may occur while the host is still a nymph. In most cases where the orientation of the cephalothorax has been observed, e.g., in Stylops, Xenos and Elenchoids, it was found that the side of the cephalothorax in contact with the host body is the dorsum, the venter being the outer, exposed side. However, Saunders 1853 found the reverse to be true in Stylops sp. and Hylecthrus sp. and Kirkpatrick 1937 ab. showed that Coreoxenos also normally lies with the dorsum as the exposed side. In each species the orientation of the 2 sexes is identical. This difference in orientation among the different species is most likely correlated with the position of the larva in the body of the host. In Coreoxenos, the mature larva lies with its venter against the body wall of the host, and in effecting emergence the cephalothorax is curved ventrally so that when the process is complete the body is U-shaped, the dorsum being outward with respect to the host body. In other species for the inverted position to be taken the larva must assume the same internal position with respect to the host body wall, but they lie with the cephalothorax and abdomen in the same plane and effect extrusion by a series of forward thrusts. Female Decerotochera undata Pierce is found in the latter position. The body is straight and cylindrical, and the first abdominal segment extends forward beyond the apex of the cephalothorax. Clausen 1940-1962. Among Strepsipterans developing in Hymenoptera, the cephalothorax of the parasitoid is extruded through the intersegmental membrane of the abdomen at any point on the periphery, although a greater number are found dorsally than ventrally or laterally. The natural curvature of the abdomen of the bee or wasp, with the dorsum decidedly convex, allows for more ready extrusion in that area. The cephalothorax of the parasitoid is always directed toward the rear, a position induced by the overlapping of the host's abdominal segments. Those of the males are usually found overlying the third segment, while those of the females are generally on the fifth segment. However, they may be found on any of the other segments with the exception of those comprising the genital system. The point of extrusion of the cephalothorax of the species attacking Hemiptera and Homoptera seems to present a greater consistency within each species than is the case with those infesting Hymenoptera. The cephalothorax of both sexes of Coreoxenos is extruded dorsally through the intersegmental membrane between the third and fourth abdominal tergites. That of the male if found on the median line, or close to it, while that of the female is most often found near the lateral margin. See Clausen 1940 for diagrams. Those on Fulgoridae are regularly upon the pleural region only, while the species attacking Cicadelidae are found in either the dorsal or the ventral position. Perkins 1905c. Perkins also found that those of the males of Halictophagus are found either dorsally or ventrally, while those of the females are in the pleural region only. Pierce 1909, 1911, 1918 found males of Stenocranophilus quadratus Pierce to be located on the fifth segment of Stenocranus and invariably dorsal in position, while the females are on the third segment and usually lateral. In Tedigozinos orientalis E and H, most are ventral and located on the fourth or fifth segments of adult hosts and on the third segment of nymphs. Generally, it seems that in Hymenopterous hosts the point of extrusion of the females is farther back than that of the males, while in Homoptera the opposite holds. Pierce 1909, 1911, 1918 considered this condition due to the fact that female parasitoids are larger than males in the first case and smaller in the second. Pupation. In species with opodous females, the individuals of this sex transform to the adult stage directly from the seventh instar larva without an intervening pupal stage. However, the male undergoes a complete metamorphosis where the pupa develops within the seventh larval exuvi in situ in the host. It may not completely fill this puparium, and in some cases the body is withdrawn from the cephalothorax so that it lies entirely within the host body. The adult male may remain within the puparium for several days after casting the pupal skin, and it then effects emergence by pushing off the operculum or head portion of the cephalothorax. This occurs during early morning hours, especially in bright sunlight. The host dies prior to this time, but this does not affect the parasitoid, for adult males have been reared from hosts that had died nearly a week previously. Clausen 1940-1962. Parker and Smith 1933b, 1934 found in Eoxenos and Silvestri 1933 in Mengeniliquae silv, which have free-living adults of both sexes, that there occurs what was considered to be a pupal stage of the female as well as of the male. The pupa of Eoxenos is found within the last larval exuvi, and at emergence of the adult the pupal skin is left almost intact. Sometimes the female remains permanently within the larval exuvi, in which case the pupal skin is torn into fragments and pushed back into the posterior end. The larval shell remains intact except that the head is detached, and the opening thus made serves as a means of exiting for the triungulinids. Males also pupate within the last larval skin, and at emergence of the adult the head and first thoracic segment are broken off. The pupal shell is usually found within the larval exuvi. Clausen 1940-1962. Mating. Because males are so scarce in Strepsiptera, it has been thought that reproduction is unisexual in most species. The activities of the males during their brief adult life, usually less than 1 day, is of interest, because they are greatly attracted to the host insects, and the finding of a host is followed by a search for the female parasitoid. Attempts at mating were noted in several species, and Pierce 1909, 1911, 1918 noted several instances where this was seemingly successful. Smith and Ham 1914 concluded that fertilization cannot take place, however. Their evidence supports the claim for unisexual reproduction based on 1. That there is no opening of apparatus in the female adapted for conveying the spermatozoa to the eggs, and 2. The eggs remain throughout their development encased in the follicular epithelium of the ovary, so that access to them by spermatozoa which may have entered the body cavity is difficult. 3. Parthenogenesis must occur as a normal rule in parasitoids of Halictus where males are extremely rare. 4. The known stages in the polar body formation of stylops are inconsistent with the view that fertilization by a spermatozoan has been effected, and 5. Actual copulation by the male has never been adequately observed. Copulating pairs of Esoterema were observed by Perkins 1918b. A close examination after killing pairs revealed that the oedagus was inserted into the brood chamber. Hoffender 1923 found stylops in copula on a female of Andrina flavipes perez for 2.5 minutes. Such observations are nevertheless not conclusive, and it was not until Schrader studied X. wheeleri that the apparent mating, as observed by others, was proved to result in egg fertilization. Mating was observed in 10 cases and over a period of 20 to 50 sec. In each case females were of a certain age, or stage of development, for the cephalothorax had been excerpted only 4 to 5 days. This partially explains the difficulties experienced by other earlier investigators, who may have been using older females that had already been mated. The spermatozoa are released through the genital opening into the brood canal and enter the body through the 4 trumpet-shaped ducts on the abdominal venter. They then disperse throughout the body, penetrate the egg membrane, and effect fertilization. A cytological examination of virgin females showed that the eggs develop as far as the metaphase of the first maturation division, which is reached 4 to 5 days after extrusion of the cephalothorax. This condition is maintained for 10 to 14 days, after which degeneration occurs. Thus, it was demonstrated that not only does fertilization take place but that parthenogenetic reproduction cannot occur in this species. In species attacking Halictus, mating may take place in the autumn and only the gravid females persist until the following spring. Collections at this time would suggest that the entire population is exclusively of the female sex. Extended observations were made by Kirkpatrick, 1937 AB, on the mating behavior of Coreoxenos. The male is strongly attracted to the host insect, even if it is unstylopized, and is not at all attracted to females of its own species after they are removed from the host. It apparently is attracted first to the host by sight and then to the female by touch. To mate, it is first necessary for the male to penetrate the membrane covering the exposed lateral genital opening. In Eoxenos, Parker and Smith noted that the edagus of the male apparently penetrated the body all along the median ventral line rather than the genital opening on the seventh segment. Females remain capable of being fertilized over variable periods. In Coreoxenos, mating may occur successfully 4 to 7 days after extrusion of the cephalothorax, but in one case a female kept in confinement was successfully mated 119 days after extrusion. Life cycle. The life cycle is exceeding variable, especially in species which attack the host in its immature stages but do not themselves reach maturity until the host attains adulthood. This applies in particular to those attacking polistes and other wasps of similar habit which are subject to attack throughout the larval period and to species having pentatomid hosts which are attacked in any nymphal instar but mature only in adults. The pupal stage of the male of X. vesperum rossi takes 28 to 32 days, Nasanov 1892, 1893, while Saunders, 1853, found that only 8 days elapse from the extrusion of the cephalothorax to emergence of the male of Hylecthrus rubiscens. In E. labobene, the pupal stage is finished in 12 days. Males of C. antestia complete the cycle from entry of the host to adult emergence in a minimum of 50 days, of which the pupal stage represents ca. 12 days, while the female attains the adult stage in 34 to 36 days, of which 28 to 30 days represents the time from entry of the triungulinid to the extrusion of the cephalothorax. Clausen 1940-1962. Mated females of those species attacking bees and wasps usually hibernate. However, Hylecthrus is believed to overwinter in the early larval stages within the Prosopis larva, this difference being made necessary by the host's hibernation, which occurs in the larval rather than in the adult stage. In Eoxenos there seems to be generally only one generation per year. Adult gravid females are found during autumn and conceal themselves under stones and other objects during winter. Embryos do not develop in their bodies until the following spring, and the triungulinids leave the parent body early in July. Triungulinids are adapted to a relatively long period of free life, which is required to allow them to reach their hosts. Coreoxenos survives for a max of 19 days in the absence of hosts and 34 days if external attachment to the host occurs. Most of this longer period is passed in an entirely inactive condition on the host body while awaiting the molt, and this seemingly accounts for the greater longevity. The first molt occurs 3 to 6 days after entering the host, and a corresponding internal period of 1 week or less has been found in other species. Clausen 1940-1962. The mature larval stage is attained in Hymenopterous hosts only when the host is nearing the time for transfer to adulthood. Therefore, it is probable that the histolytic processes associated with pupation provide the stimulus for completion of growth. Clausen 1940. Developmental delays until this host condition is attained is known to be obligatory in other parasitic insects. The cephalothorax of X. vesperum is extruded from the host abdomen 5 to 8 days after the polistes adults emerge from their cells. In Ophthalmoclus sp. and Stylops spp, adult males usually emerge the day hosts first leave their nests. This does not, however, aid much in determining the time of extrusion, for the hosts may have been in the adult stage in their nests for a considerable period. The cephalothorax of X. pallidus bruce may not be extruded until several days after the wasp has left its pupal cell. Pierce 1909, 1911, 1918. Extrusion in Coreoxenos occurs not more than 36 hours after the final transformation of antestia. Saunders 1853 recorded the activities of male larvae of Hylecthrus rubi underneath the abdominal integument of fully developed Prosopis pupae, and extrusion took place almost immediately after the host had assumed its adult form. Clausen 1940 believed it logical for extrusion to take place very soon after the pupal skin was cast, for the integument is more easily penetrated at this time than when it has hardened. Sex ratios of host and parasitoid. Pierce 1909 provided an extended discussion relative to sex ratios of several Hymenoptera that were stylopized, the percent of each sex stylopized and the sex ratio of the parasitoids. From 1,553 Polistes annularis L collected during October, only 15.6% were female. Of this number, 17.2% were parasitized, but the percent was 19.7 among males and only 2.8 among females. Sex ratio of Xenos pallidus favored males by about 1:2. In Andrena crawfordi vir, 43.3% of 266 bees collected were female, and of these 53.1% were parasitized compared to only 20.6% of the males. The sex ratio of the parasitoid favored females slightly. September rearings of Xenos wheeleri from Polistes gave a majority of more than 2:1 favoring males, while May and June collections yielded female parasitoids only. Schrader 1924. Earlier records of the same species taken during late August gave males in excess by 4:1. Wheeler 1910. Apparently not only does the sex ratio of the host vary widely among seasons, but the ratios of the parasitoids differ markedly during the year and in different hosts. Male Polistes do not survive winter well, and few of those of either sex from which parasitoids have emerged would survive. Stenocranophyllus quadratus males predominated in a ratio of 1.7:1, and parasitization of female Stenocranus was a bit higher than of males. Clausen 1940. Females of Alentinus japonicus, parasitic in Delphicodes, predominated 2.5 1. While in Pterygoxenos orientalis, parasitizing Parabolocratus, the sexes were about equally abundant, Asaki and Hashimoto 1931. The greatest number of individuals that are able to develop in a single host is about 31 larvae of xenos reported by Bruce, 1903, 1905, in 1 larva of Polistes. The highest number to have reached the excerted stage was 15, these being male pupae found in a male of P. annularis. However, regularly the number in each host ranges between 1 and 5. Asaki and Hashimoto 1931 found 1 to 3 in each individual Homopterous host, and Sebramanium 1922 studying Pyrroloxenos compactus found 1 to 3 in Idioceras sp. A maximum of 7 was recorded by Misra 1917 for this species in nymphs of Pyrrilla aberrans Kirby and 12 in the adults. Bruce 1905 concluded, as a result of studies on several species of xenos, that there is a tendency for all parasitoids developing in a single host to be of the same sex. In X. pallidus parasitizing Polistes annularis, of 100 male hosts containing more than one parasitoid and averaging 2.99, 26 lots were males only, 11 were females only and 63 were of both sexes. Pierce 1909. This disparity between the number of pure colonies of the two sexes may be explained by the predominance of males in the sex ratio of the parasitoid. Wheeler's studies on X. wheeleri showed an average of 2.4 parasitoids developing in Polistes sp., the maximum being 11. Wheeler believed that in all instances where the number was in excess of 4, all of them were of the same sex. This is suggestive of a differential death rate among larvae of the both sexes, the males dominating because of their smaller size and lower food requirements. Clausen 1940. Studies on the same species by Schrader 1924 supports Wheeler's contention regarding the differential death rate among larvae, and dissections of larvae, pupae, and adults of Polistes showed that the larvae of the two sexes of xenos were present in about equal numbers. Figures for X. vesperum in P. gallicus L presented by Vandal 1932 show that 18 out of 31 wasps containing more than one parasitoid bore only one sex, of which 16 were male broods. In 13 mixed broods, 9 had an excess of males, the numbers were equal in 3, and only 1 contained an excess of females. However, 21 out of the 25 solitary individuals were males. Limited observations on Ophthalmoclis sp. in Specs by Peel 1933b showed that males were the minority in mixed broods. Detailed information on sex ratio in studies of Corioxenos, of which 1 to 5 develop per host, show field collected material numbering 1,000 hosts had 44.7% with only a single parasitoid, among which females predominated in a ratio of 1.5, 1. Clausen 1940 considered this to be the normal sex ratio of the parasitoid. Where hosts contained 2 parasitoids, the female preponderance was higher, the ratio being 2.4, 1. With a greater number in each host, the sexes were present in about equal numbers. Such records show no tendency for the brood in a host to be of only one sex. Of 249 antestia containing 3 or more extruded Corioxenos, 16 of the broods were 100% male, 9 were 100% female and 224 were mixed. Stylopization effects on the host. Many noticeable changes are brought about by Strepsipteran parasitization, among which are included various internal changes due to the feeding of larvae and often a pronounced influence on the primary and secondary sexual characteristics of the adult host. Most seem to result from nutritional balance upsets of the host through feeding by the parasitoid. Early studies on the effect of stylopization were by Perez, 1886, Pierce, 1909, Wheeler, 1910, Smith and Ham, 1914, Perkins, 1918a, and Salt, 1927a, 1931a. Stylopization affects various bee and wasp hosts by either accelerating or retarding development to the adult stage. Westwood, 1840, noted that parasitized Andrena adults appear ca. 1 month earlier in springtime than do healthy adults, and Saunders, 1853, observed earlier emergence in the case of Prosopis. Pierce, 1909, recorded that the first adults of A. crawfordi to be found in the field were parasitized by 59-79%, while those taken a few days later showed a much lower percentage. Only adult xenos were found in Polistes wasps of the autumn broods, which were considered to belong to earlier broods that had been retarded in their larval and pupal development. Wheeler, 1910. Such results indicate that there is only a single parasitoid generation annually, though Schrader, 1924, demonstrated that there are really 2. The latter found that the host larvae and pupae which remain for a longer period in the nest are the most heavily parasitized and that their random distribution in the nest indicated an actual retardation. It thus appears that there is an actual acceleration in development among the bees in contrast to a retardation among the wasps. Clausen, 1940. Studies by Salt, 1927, show that because of the presence of the parasitoid in the body of the host larva and its continuous abstraction of food materials from the blood, the host is kept in a continuous state of hunger. Thus, stylopized bee larvae feed more rapidly and consume the food supply in the cell earlier than unparasitized individuals. They then pupate and emerge at an earlier date than do the unparasitized individuals. However, in Polistes the food supply is not of limited quantity, and the larvae are thought to be provided with food as long as they need it. Parasitized individuals have the same persistent hunger as is found among the bees, but are able to continue to feed enough to compensate for the materials taken by the parasitoid. This leads to the larval stage being prolonged, and adult emergence is correspondingly delayed. In Fulgoridae, which are able to secure whatever added food is required, the parasitized individuals likewise show a retardation in development. Parasitism by male Strepsiptera seems to result in greater injury and causes more profound changes in the host than does that by females. Many of these changes occur while the host is in the mature larval and pupal stages, and thus the abstraction of large quantities of food materials from the body for the production of eggs by the parasitoid female is not involved. Clausen 1940. When parasitized adults emerge, the male parasitoid is a more highly organized body than the female and has been more of a drain on the host's vitality. However, this explanation is not in accord with that given for the preponderance of males in heavily parasitized hosts, which is attributed to a differential death rate. A general marked loss of vitality occurs among parasitized host adults, ranging from merely reduced activity to an almost complete cessation of normal functions. Parasitized male bees and wasps have been found to mate normally, which also is occasionally the case with females. The females among these hosts seldom attempt to carry pollen or to build and furnish nests. Some species of Andrena when parasitized never carry pollen, while others and Chlorolictis also, may do this frequently or occasionally. The host often sustains severe injury to its internal organs, which is caused by the removal of food from the blood rather than by direct feeding and tissue laceration. Atrophy of the ovaries is so far advanced so that mature eggs cannot be produced. Kirby 1828, Clausen 1940. The secretory and poison glands of Andrena are reduced, the tracheal system is reduced and with the vesicles few and imperfect, the nerve ganglia of the abdomen are atrophied, and the intestine is empty and dislocated. Newport 1845-1853. The male reproductive system seems unimpaired, though Perez 1886 mentioned that these organs are atrophied only on the side bearing the parasitoid, and Salt 1927 noticed a slight size reduction. However, in Antestia spp. Parasitized by Corioxanose antestiae, most males prove to be sterile as determined by breeding experiments with unparasitized females, even though apparently normal spermatozoa were present and mating apparently occurred normally. Functional sterility may be more general than is obvious, however. Among females that are parasitized at the time of the final molt and that contain only a single parasitoid, a small number, ca. 1%, contain 5 to 15 apparently mature eggs each. However, despite this, no female ever oviposits. Hosts do not always die as a result of Strepsiptera parasitism. Death of parasitized leafhoppers usually follows soon after emergence of the male parasitoid, this being due to the empty puparium providing a large opening to the body cavity through which desiccation of the viscera occurs and which also allows disease organisms to enter. Perkins 1918a, B, Misra 1917. Female leafhoppers with male parasitoids merely become sluggish at the time of triungulinate exiting and may live for a considerable time. Perkins believed that the presence of a fungus disease was essential to maximum effectiveness of these leafhopper parasitoids. Because of the injury to the reproductive system of the female hosts, this seems to be inconsequential, for reproduction has ceased and these individuals cannot contribute further to population increase. In Antestia spp, adults containing either male or female Corioxanose live almost as long as healthy individuals. Adult Pholistes bearing empty male puparia may live for a considerable time and may even be able to hibernate successfully. Clausen 1940. The first extended study on the external changes brought about from Strepsiptera parasitism was by Perez, 1886, on a large number of Andrena spp, with other researchers following. See Clausen 1940. Apart from mechanical distortion and occasional direct injury induced by the extrusion of the parasitoid cephalothorax, the general external changes are evident in the relative size of the body parts, integument, puncturation, pilosity and wing venation. The principal secondary sexual characters that are affected, with the direction of the changes that take place, include antennae color and proportionate length of segments, pubescence color, pollen collecting apparatus, size of tarsi and number of bristles, clypeus color and shape, mandibles color, and genitalia size, shape and proportions of parts. Clausen 1940 60 seconds. In Pholistes, no great modification in secondary sexual characters results from parasitism, but the most marked changes are found in Andrena. Perez concluded that the modifications in the latter constitute actual inversions of development. The stylopized Andrena, male or female, is not merely a diminished male or female, it is a female which takes on male attributes, a male that takes on the characters of the female. Other researchers later corroborated this. Such changes are so pronounced that in many instances parasitized individuals have been described as new species. Clausen 1940. There is no uniformity in the effect of stylopization on the external characters of Hymenopterous hosts. Even among individuals of a single species, the differences may be very large, probably due to a variation in the stage of development at which parasitization occurs. In some cases, particular effects are revealed in only one sex, and these differences are correspondingly greater between species and genera. Such various changes occur in a definite order. Salt 1927. Asaki and Hashimoto 1931 found among Fulgoridae a marked neutralization of the genitalia of both sexes of Delphicodes fucifera horf, resulting from parasitism by Elentinus. However, no such effect was found in the cicadelid, Parabolocratis prasinus mats, parasitized by Tetigozinos. No changes in secondary sexual characters were observed in the pentatomid Antestia sp. When attacked by Corioxanose, however. Kirkpatrick 1937 ab.